Feeding patterns of the round goby and implications on population control of invasive freshwater mussel species by Nick Brown, Mallory Deschamps, Hilary Nickel, Caitlin West, and Alexis Zaborniak. In Lake Erie, the fishing industry is a major source of income for the surrounding community. However, the industry has taken a turn in recent years due to harmful algal blooms along with the introduction of invasive species. During the warm months of summer, agricultural and industrial runoff inputs excess nitrogen and phosphorus into the Lake Erie watershed. This process is known as eutrophication. As a result, algae proliferates at a very high rate that exceeds consumption. The layer of algae becomes so thick that it prevents light from reaching plants beneath the surface, thus lowering oxygen production in the hypolimnion. This results in anoxic waters that kill cool water hypolimnion fish. Because of the lack of mixing of the oxygen-rich epilimnion from algal production, oxygen is not distributed to below the thermocline. In addition to the harmful algal blooms, invasive mussel species, mainly the Dresenid family of mussels, are outcompeting native mussels. In fact, zebra and quagga mussels attach to the hard surface of native mussels, reducing their ability to thrive. Therefore, the food web is being drastically interrupted, and Diphtheria populations, which are shrimp-like species that many fish rely on for food, have declined greatly. As a result, Dresenids are contributing to the economic downfall of the fisheries. Currently, many studies have investigated the feeding patterns and viability of a fish that we believe may be able to be supplemented into Lake Erie to decrease invasive mussel species while also maintaining native mussel species. This fish is called the round goby. After much research, we hypothesize that the round goby will demonstrate a size preference for mussels that they consume when offered a choice between larger native mussels and smaller invasive mussels. After the course of a short study with 3 test groups, which will be gobies with invasive mussels, gobies with native mussels, and gobies with a choice of both, an ANOVA may demonstrate a difference in mussel populations between the 3 test tanks. This may implicate that gobies, if supplemented into Lake Erie, could selectively reduce Dresenid mussel populations without harming native populations. However, we must extensively research the feeding patterns of the round goby, as well as its viability and effect on native communities. The following articles will demonstrate such research. An invasive species is a species that is not native to the environment and poses drastic environmental change upon its inoculation. Such a species is the round goby. These changes in the environment that an invasive species introduces may be positive or negative. It is imperative to investigate these changes, as round goby supplementation into the Great Lakes may result in a decline in harmful invasive mussel species, but could also have harmful effects on other species as well. As such, it may not be the best candidate to solve the current imbalance in Lake Erie. Oddly enough, invasive species are quite unique, although they tend to occupy an ecosystem similar to their native one. For the round goby, their native home is the Black and Caspian Seas, and their invasive home is the Lake Erie watershed. Invasive species such as the round goby are successful in non-native environments if they have genetic plasticity, thus allowing them to adapt to non-native environments. Once adapted to a new environment, invasive species can have great impacts on community populations. For example, decreased invertebrate species were observed in goby-infested tributaries of Lake Erie when compared with goby-free streams. This goes to show that gobies may have a significant impact on reducing the invasive mussel populations when supplemented at greater populations into the lake. Although invasive species occupy a similar environment, they can play a very different role when interacting with a different community and finding their realized niche. When choosing a species to decrease harmful invasive mussel populations, such species should be viable in the new environment as well as cause little harm to other species. As a result, the effects of round gobies in a non-native ecosystem need to be investigated. This is exactly what Coronis et al. aimed to do as they investigated if biological characteristics affect round goby invasion success and if invaded communities are affected by the round goby. When the study first began in 2007, sampling sites were chosen from Wisconsin tributaries based on a 2007 census. All sites were wadable, meaning they were less than a meter deep, and they were downstream from barriers such as dams. Researchers noted 30 goby present sites, 52 absence sites, and 5 invaded sites. For the monitoring portion of the study from 2007 to 2010, 23 of the 30 goby present sites were chosen to be monitored from the same tributaries. All of these sites were also wadable and contained communities of both round gobies and at least one of the 5 native benthic fish species. Native fish species were chosen based on past studies that suggested negative effects of round gobies or because of a diet overlap. Fish were captured using the electrofishing method, which is pictured on this slide. Specifically, researchers used a battery-powered backpack with a pulsed DC current. Each site was electrofished for 20 minutes while researchers worked upstream, sampling from bank to bank. All captured fish were identified, measured, inventoried, and released. However, round gobies were kept and euthanized using a euthanasia solution. CPUE, catch per unit effort measured in individuals captured per minute, was used to determine species density. In addition, round goby density was measured in four rivers, 3 sites per river for a total of 12 sites as part of this study. However, a towboat electrofishing unit with gas-powered pulsed DC current was used instead of the battery-powered backpack. This offered a greater strength current. Each site was sequestered using 2 fishing nets, and CPUE was again determined per the same method above. Some environmental variables such as watershed area, watershed slope, watershed soil permeability, average July air temperature, base flow index, and stream gradient were measured using a geographical information system database. A couple of statistical tests were used to analyze the data collected. One of these methods was the redundancy analysis, known as RDA. This associated the environmental conditions I just listed and the round goby CPUE. The RDA allows this information to be expressed in a linear fashion. Rare species and tube-nosed gobies belonging to the same family as round gobies were excluded from the analysis. Furthermore, an R statistical package was used to perform a redundancy analysis, correlation analysis, and linear regression.
This table shows the Shannon diversity, number of taxa present, and CPUE for fish species in goby present and goby absent streams. Looking at the mean for the Shannon diversity of the goby present streams and comparing the value to the goby absent streams. This is showing that there is a greater diversity of species when there are round gobies present. Also, if one looks at the number of taxa present in each category, it is shown that there is a larger number of taxa where there are gobies present. The overall results of this journal is that native fish species were not affected by the invasive round gobies, which was originally thought. The first graph shows the percent of gobies relative abundance of all individuals captured in an area on the x-axis, and the frequency that this percentage was calculated for each area on the y-axis. This shows that even though the round gobies are present in most areas, they only made up 0 to 10% of the total fish captured. The second graph shows the round goby catch per unit effort, or CPUE, which shows how many fish were captured per minute. The frequency on the y-axis shows that in most areas there were less round gobies captured per minute, showing that in most streams there was a low density of the fish. Through looking at the results, it is shown that the round gobies are not necessarily positively affecting the other species that are present because there are other factors such as food source and temperature, but the fish are also not negatively affecting the other species present, including the abundance of native species. In fact, researchers conclude that species negatively associated with round gobies prefer conditions that are not optimal for the round goby, thus making them unlikely to co-occur. Results from this study showed that, oddly enough, round gobies do not decrease species richness as most invasive species tend to do. However, favorable watershed areas correlated with increased habitat complexity and resource availability may be the underlying reason for the maintenance of diversity. This is important to our research because it shows that the invasive round gobies are not going to decrease the amount of the other fish present as long as the environment characteristics favor a high diversity of fish species. Furthermore, the study showed that in the 3 years the study was conducted, the abundance of gobies increased from 19 to 23, showing that the gobies are a viable species for the Great Lakes. This is critical in that the survival of round gobies must be high in order for supplementation to be sustainable to counteract invasive mussel species. While this specific study was conducted in the tributaries feeding into Lake Michigan, this study can be applied in Lake Erie tributaries to receive the similar results if environmental conditions are similar.
The Great Lakes have grown considerably since 1990, and an estimated 9.9 billion fish in Lake Erie were documented. As the Lake Erie population have expanded, studies have documented round gobies migrating into tributary streams, which are pathways to inland lakes and reservoirs. In the summer of 2008, round gobies were present in 50% of Lake Erie's tributary streams, which also had reduced macroinvertebrate species and mussels. There is little to no data on the activity of round gobies during the colder months. Therefore, round gobies are thought to have seasonal migration patterns, moving deeper offshore during winter and residing near shore during warmer months. There are 3 objectives to the research done by Panuto: determining if round gobies were present in tributary streams throughout the year, assessing round gobies' seasonal size distribution, and documenting round gobies' prey preference and energy consumption. This research is pertinent because resource managers want to know whether round gobies are using tributary streams as reproductive areas or corridors to inland waters. It also reflects round goby competition with other invasive species. Monthly samplings were conducted upstream at Big Sister Creek and 18 Mile Creek in New York between April 2006 and February 2007. These sample sites were 1 and 2 kilometers from Lake Erie. 100 meters were sampled at each stream using an electroshocker for up to 1,000 seconds or until 50 round gobies were collected. The rocks at the bottom were shocked, causing the large rocks to lift, which allowed the current to wash stunned gobies into the nets. The length of the round gobies and the monthly catch per unit effort were compared using a 2-factor ANOVA without replication. In July 2007, 25 round gobies were collected 15 kilometers upstream from Niagara River. The digestive tract of each goby was identified using a microscope. The energy consumption by round gobies was estimating using a calorimeter. Invertebrate samples were added to the calorimeter for estimating the biomass of each gut over the last 8 hours. To provide an estimate of round goby impact on stream energy flow per day, the value was multiplied by 3 to obtain a 24-hour value. This table shows the values collected for 3 creeks in the eastern basin of Lake Erie. At these 3 locations, the physiochemical conditions were similar. The water temperature, dissolved oxygen, and conductivity levels were similar in all streams in all months. Turbidity levels in the streams were highest in the spring and decreased in the fall. There were no recapture events during the field study, which prevented estimation of population size in the streams. The 3 streams were relatively the same width, depth, and speed. It was documented that Big Sister Creek had more plant canopy cover than the other 2 creeks. 18 Mile and Big Sister Creek were sampled on a monthly basis between April 2006 and February 2007. Peak temperatures occurred in July in both streams and were lowest in January before it rose again. The total catch of round gobies increased from April to October in both streams. Round gobies were also more abundant in Big Sister Creek in September to October. The monthly catch per unit effort of round gobies in 18 Mile Creek was double the maximum value in Big Sister Creek. In both streams, the population of round gobies was maximized in October. However, values decreased when temperatures hit 15 degrees Celsius in 18 Mile Creek, but in Big Sister Creek, they did not decline until 5 degrees Celsius. The seasonal sampling data showed round gobies decreased in abundance with seasons and were not available for sampling in the winter. This phenomenon could be due to several outcomes, including outmigration to Lake Erie, the burial in soft sediment during winter, or predation. Round gobies were present in both 18 Mile and Big Sister streams in April, when the monthly catch per unit effort increased until October. The decline shows the possibility of outmigration by stream-dwelling gobies. Future research could be conducted with radio tagging round gobies and observing outmigration from tributary streams. The electrofishing done in the Niagara Stream confirmed presence of round gobies and their implication for stream energy. Round gobies may act more like nutrient and energy robbers by removing nutrients and energy from streams into the lake during the winter. Round gobies reproduce during May and August, which was documented when few small gobies were caught during those months. This means these tributaries appear to be sites of reproduction rather than corridors.
Community-level trophic dynamics, including feeding ecology, are important in understanding interactions between invasive and native species. Invasive species often have the capacity to process materials differently and consume different prey than native species, and subsequently can affect change in how an ecosystem functions. Feeding ecology studies can be useful in the management of invasive species by providing key clues on how invasive species can alter system trophic dynamics. This study analyzes the consumption choices of round goby along a depth gradient in the western basin of Lake Erie. Both laboratory and field components were used to evaluate prey selection, prey consumption, and then were used to describe diet based on dominance, frequency, and relative importance of prey items. The objectives for this study were to determine the species' richness and dominance exhibited in round goby prey in the western basin, and to determine the size selectivity of prey items when gobies are presented with varying size classes under laboratory conditions. For this test, a field study and a laboratory experiment were performed. In the field study, round goby dietary habits were conducted at 6 sites surrounding the Bass Island archipelago in the western basin of Lake Erie. Each site was chosen to represent variation in depth, coastal shoreline structure, and habitat heterogeneity. Same-size mesh and signs and otter trials were selected to reduce bias in shallow and deep-water habitat selection of individuals. Trot lines were selected to collect larger individuals from shallow habitats. Lines were checked after 10-minute sets. Each individual collected was immediately anesthetized after capture with MS-222 and fixed with 10% formaldehyde. Individual fish were separated into 2 size groups based on pivotal hinge point size, were sexed, and measured for total length. The entire digestive tract was dissected from preserved individuals, and the volume of prey item was estimated for each fish as a percentage of the gut content. To analyze diet consumption patterns, individuals from each sample were segregated by sex. Differences in diet were analyzed among the 6 sites to see if the surrounding environment substantially affected prey consumption. Prey items were grouped by taxonomic order for comparison. For the laboratory experiment, individual round goby were collected from the western basin near Put-in Bay, and quagga mussels were obtained from the Gibraltar Island in the western island by snorkel diving. Quagga mussels were measured and divided into 4 class sizes. For the experimental design, 10 individual mussels from each of the 4 class sizes were positioned hinge side up in a 28-liter aquarium. Each aquarium was filled with lake water and maintained at ambient temperature anywhere from 20 to 22 degrees Celsius. A single round goby was introduced into each aquarium and allowed to feed on mussels without replacement for 5 days. After 5 days, each round goby was removed from the aquarium euthanized using MS-222, and measured to the nearest millimeter total length. This table shows a condensed version of the most common prey found within the intestines of the round gobies. These results are an accumulation of data from 4 of the Great Lakes and a couple of their tributaries. As shown, for most of the year, the round gobies prefer to consume the dracenid mussels, followed by Daphnia, also known as the common water flea, and cryonomid larvae. The high consumption rate of the dracenid mussels makes the round goby a good candidate for controlling the rapid spread and growth of the dracenid population. For this graph, the total length of the round goby is measured in centimeters, and the predation index was calculated using the number of mussels consumed and in which of the 4 class sizes they fell into. Round goby individuals consumed, on average, more quagga mussels from the smallest class sizes compared to the other class sizes. There is a greater selection of mussels from the smaller class sizes than there is from the large class sizes. A positive correlation was also observed between the predation index and round goby total length.
This study, looking at the eating habits of the round gobies, found that round gobies selected for dracenid mussels, which is the family of mussels that have invaded the Great Lakes. This study also shows that round gobies consume a higher abundance of smaller mussels, and there is an inverse relationship between consumption and the size of the prey. These both help us to understand that the round gobies would be an excellent species to control the invasive mussels in the Great Lakes, specifically Lake Erie.
Our proposal is to look at the interaction between the round goby that is known to ingest certain mussels and both the native and invasive dracenid mussels that currently inhabit Lake Erie. The idea behind this would provide a possible opportunity to control the expansion of the invasive dracenid mussels while preserving the native diversity as well. Since the round goby is, as well, an invasive species, the possibility for its own expansions needs to be taken into account as well when looking at this area. These past articles show migration patterns in the round goby during the different seasons. This information is important in order to take into account that the gobies may not stay within the lake for the whole year and could change mussel populations during this time in the areas they travel to. These areas could be inhabited by the invasive mussels and could control the spread of these mussels any farther than those areas as well. When looking at the interactions between the round goby and the different mussels that inhabit Lake Erie, the preference for a certain type or size of mussel can cause a change in the goby's effect on the invasive dracenids. We would want to see that the round gobies prefer the invasive species over the native species in order to keep the populations of dracenids under control without affecting the native populations. It has also been shown that there is selection for certain sizes between the round gobies and mussels that it eats. If the gobies prefer the smaller mussels that may have not reached reproductive maturity, this could keep the population under control with less likelihood of growing out of control. Not only is the interaction between the mussels important for this research, the interaction between the round gobies and other invertebrate species that are seen in the Lake Erie ecosystem should be considered as well. This is used to show that the native species balance would not be harmed from the introduction of the round gobies into their habitat. As mentioned previously, this research proposed could be used to control another invasive species that is posing a threat to the industry that surrounds the Great Lakes. The overpopulation of mussels in the lake have overrun the beaches and could be turning away any possible tourists that could visit and partake in activities around the lake. These mussels have also affected the fishing industry by interrupting normal procedures as well as lowering populations of desired fish. Being able to control the population of mussels in the lake could prove to boost the industry around the lake as well as reestablishing and protecting the populations of native species of fish and mussels.